with what I had learned about mycelium materials. And so we gave hundreds of workshops to children, designers, entrepreneurs, teachers about different kinds of microorganisms. And then I ended up starting a PhD in 2017 at the University of Brussels. This was mostly actually to financially sustain myself, because all the previous work was done on a voluntarily basis. And with this research, I wanted to understand what it takes to valorize organic waste streams from wood and agricultural industries, while creating a solid material that is biodegradable at the end of its life cycle. So I chose to focus my attention on mycelium materials, which, as you can see on this image, are made by mixing natural fibers together with mycelium, which is the root structure of mushrooms. And mycelium will then bind all those fibers together into a solid material. Those materials have been explored already over the past few years and have led to all kinds of new applications, such as panels and bricks and leather. But due to its novelty, there is actually still a gap between the vision of using those mycelium materials in mainstream architectural applications and the actual commercialization of bulk materials. So most of the projects that we still see today are still proof of concepts. And then also I realized, and that was mostly the case in 2017 when I started with this, was that there was a lack of scientific knowledge about, for example, what kind of factors affect the biological and material properties. And that is because most of the innovation happened and is still happening within companies where knowledge is not necessarily shared within the public domain. And so that's why the scientific insights that into, for example, the biomolecular mechanisms and the interaction of the organism with the fibers on which it grows, all of this scientific knowledge forms the basis of the manufacturing process. And those insights can then be used to optimize the fabrication process. And so my interdisciplinary research united different faculties at the University of Brussels, which didn't happen before. So they had never collaborated before. And it was the group of microbiology, physical chemistry and polymer sciences, and architectural engineering. And the interaction with the master students coming from those different faculties and who worked with me over about a year on their master thesis on the topic of my PhD. Those interactions were really, really crucial because it allowed me to access infrastructure and also knowledge coming from those different disciplines, and allowing us then to link them together, which is kind of important if you're working in such in the interdisciplinary field. So there are many variables which underlie the production process of mycelium materials. I will quickly explain them in a very general way because I'm not sure if everyone is actually familiar with the production process. So mycelium is initially grown on agar plates, or in a liquid nutrient solution, or on a grain substrate. And this grain is a type of inoculation that I use the most during my research, because grains are a good starting point for mycelium to then grow on the fibers compared with the synthetic nutrients of agar and liquid inoculum. The natural fibers have to be humidified with water or with a nutrient solution. And the amount of water that is added to the fibers is usually the maximum absorption capacity of this fiber. So it will obviously be different from fiber to fiber, and you would have to test this in advance. And then the fibers are heated at a high temperature. We call this pasteurization or sterilization, to eliminate any type of already present organism in the substrates. It's a rather crucial step to prevent contaminations during the growth or the inoculation process. So the green fungus, which you see here, this is what we call contaminations. It grows much faster than the white rot fungi, and the problem is often that this type of fungus can be unhealthy for a human because it will rapidly produce spores, and it will also inhibit any form of growth from the Mycelium species. Then a specific amount of Mycelium spawn, which is usually about 10%, is added to the substrate in a sterile condition. And then we let Mycelium colonize the substrate in filter bags. And this is to ensure that the Mycelium will colonize the substrate completely and homogeneously. After a few days, we hand pack the Mycelium substrate in a sterilized mold, which has already the desired shape of the final product. So in this case, we were growing rectangular shapes. And then the mold is sealed with a breathable foil, mostly to maintain a kind of microclimate inside. And this is all done in a controlled environment. The Mycelium can be grown in different phases. It really depends on what you're doing. But first, we grow it in this rectangular mold to bind the fibers together. And then in a second stage, the material can be harvested and grown a few more days outside the mold, mostly to solidify the outer skin of the material. I don't know if you can see it on the image, but the fibers are still visible, and this is because there was a wooden panel preventing the access to oxygen, and therefore the Mycelium couldn't grow well on the outside. But in 2 or 3 days, this material is then fully grown on all sides, as you can see here. And then the material is heat treated at a temperature usually above 60 degrees for several hours to end the growth process and to dehydrate the material. So at this point, the material is made inert. Basically, the Mycelium organism is dead. And then different kinds of coatings or post-processings can be applied to the material to improve its properties. And it will then result in a solid material that is biodegradable in the soil at the end of its life cycle, making this a fully circular process. So back to my PhD. The main goal of the research was to explore the principal factors that affect the biological and material properties of Mycelium materials, and then also to broaden the potential of new architectural applications with fungal organisms. So it was a very hybrid investigation across many disciplines, mostly aiming at contributing to this growing field of different kinds of manufacturing processes with Mycelium. So ultimately, it was, I think, a very comprehensive overview of the crucial aspects that come into play when we want to produce this type of material. So I developed a method for selecting fungal species that incorporate biological, chemical, and mechanical performance criteria. I also investigated the interaction between the fungi and their feedstock and the material properties of different feedstocks. And then I studied whether it was possible to optimize some mechanical properties with different additives. So I tried with bacterial cellulose and with nanoclay. And then the second part of the research was about novel fabrication processes to produce large-scale architectural formwork. And finally, different additive fabrication and design strategies were explored with 3D printing. And so initially, my work started from an engineering point of view. I really was trying to navigate between, on the one hand, having wanting to come up with a solution for urgent problems, like replacing polluting existing construction materials, such as EPS, with mycelium materials. And more towards the end of the research, unfortunately, the funding was then almost finished, I started to explore the living capacity of mycelium, because okay, the promise to realize a shift towards biofabricated material is already thrilling in itself, but the concept of using systems that would remain alive during the entire lifespan of the material would open up totally new perspectives in which the material have smart functionalities, functional properties such as environmental sensing, adaptation, or it could self-heal and have a self-healing capacity. There are many challenges when we want to grow mycelium materials. I won't talk about all of them, but I'll pick some of the things out of different projects on which I worked to make my narrative clear towards why I'm now working with living materials. And so, as I said before, one of the problems that we are facing when we grow mycelium materials in molds is that there is a lack of air, which will significantly restrict the viability of the fungal organism. So this image makes it really clear that mycelium is unable to grow on the bottom there because there is a lack of oxygen. And actually, when we think about it, this kind of molds will also restrict the complexity of the final product in terms of geometry and about scale as well, especially on the construction scale. And then the other thing is that when we dry the mycelium material, the organism is heat killed, and therefore it is removed from any biological functionality, such as its capacity to regenerate itself. And so that's why we wanted to investigate the potential of keeping the mycelium organism alive during the entire fabrication process. So we had to develop a method to create a large mycelium block. So like what I said before, it's unable to grow properly inside the material when there is no oxygen there. And then afterwards, we used an on-site robotic wire cutting technique to shape a living block of mycelium material. And so after the wire cutting, the two pieces of the living block, as you can see on the left image, they were reassembled. And while the organism continued to grow, it produced this white, grayish skin all around the exposed fibers. And by increasing the humidity during the last incubation phase, we allowed the heath, the mycelium, to bridge the gap of about 5 millimeters between the two elements. And as you can see on the most right image, those two pieces would then fully reconnected. And the prototype kind of demonstrates how we can use the potential of living organisms to bioweld. The fungal organism had survived the uncontrollable environment during the transport. So the mycelium blocks were made in Belgium, but they were wire cutted in Denmark. So they were sent just by normal post to there and then sent back to Belgium to finish the growth process. And the wire cutting facility was an unsterile environment. And I was actually quite surprised that it wasn't contaminated at all, although it had been in those unsterile environments while being still alive. And it had also, the blocks had been severely damaged because we had cut off the surface, the edges of the blocks, and we had cut off this shape, this curved shape. And even with all of this, it was still capable of regenerating itself. There are a lot of other challenges which come into play when we want to keep mycelium alive. One of them is that it will create fruiting bodies, which is often not really wanted in building materials. And while the organism continues to grow, it will produce a denser network, as you can see on this image, the microscopic image here on the left, well, on the right side, which it's a good thing that it will produce a denser network, because then it's able to hold the fibers even more compactly. But the other problem is that the mechanical properties will decrease over time as the organism continues to grow, and it therefore also continues to degrade the natural fibers. So keeping mycelium alive in a composite material, which is composed of natural fibers, seemed like a challenge to us, unless the purpose of the material is to degrade. And that's what we wanted to show off with this prototype, where we set out to investigate whether the traditional fabrication methods of mycelium materials could also valorize plastic waste. So we injected a bioremediation process of plastic waste into this living material with an architectural purpose. And so the goal was to and it happened actually, the plastic waste particles which were combined with wooden particles started to fully degrade. Then scaling up the production of living mycelium materials, it's giving us a whole set of other challenges. And at the HBB in Newcastle, we have an experimental building. It's a building we call the OHM, and where we can test those kinds of questions. So we work with a whole range of microorganisms making bacterial cellulose, making biocementation, and also mycelium materials. And one of the prototypes we made here is called Bionite. It's a freestanding architectural construction, which is made using knitted fabric, mycelium, and bacterial cellulose. And it addresses how digitally programmed knit can be used as a shape agent for the in situ growth of mycelium, and then in a second stage, the addition of bacterial cellulose sheets. So the knitted textile contains programmed tubes in which we inserted mycelium materials. We developed an extrusion technique which enabled us to fill the knitted tubes, and which also reduced the distortion of the knit, which was caused by the bulk substrates. So the kind of paste we developed, it behaves like a mycelium mortar or mycelium cement concrete. It will stiffen up the textile after it is grown. And this Bionite was grown in a dome shape, which was then turned after the growth and after the drying. And as you can see, the assembly of the structure happened again in a not very sterile conditions, because it's simply not possible at that scale. And we did expect again to have contaminations from environmental fungi on the knit during this in situ growth. So we systematically removed it when it happened. And then the optimized curvature of the vault was achieved by exploiting the textile's behavior in tension. So the prototype was formed by hanging the structure in a soft state to form this kind of catenary hammock. And then afterwards, it's consolidated thanks to the mycelium growth. And at the final stage of the fabrication, the structure was rotated with 180 degrees to form the catenary vault. And before doing that, we had a lot of concerns about rotating the structure because we were afraid that it would not be able to withstand the bending forces which act when you turn such a structure. But the combination of using a compressive material, such as mycelium materials, and then the tensile strength of the knitted fabric meant that there were actually no problems at all during the rotation. And so at this stage, this prototype is entirely self-supporting. Filamentous fungi are also a promising resource for the fabrication of leather-like biomaterials. And recently, there has been a lot of progress in a kind of system-level understanding of genetic in combination with a lot of developments in synthetic biology engineering approaches. And that has given rise to the emergence of engineered living materials, ELMs. And so in research in the literature, we see a huge promise in those engineered living materials. But despite all of that, we haven't seen examples yet of fully biological living materials. So most of the research has until now focused on combining, for example, yeast or bacteria with an inorganic material. Or what you can see here on this slide is they have focused on growing an organic material, which is bacterial cellulose, but the structural component of the material is the cellulose and it's not the bacteria itself. So the goal of the current research is to develop a biological engineered living material that is structural at the meter scale and which is not a hybrid. So we are talking about pure mycelium material, not combined with any kind of fiber or additive, and which has functional properties such as its self-healing capacity. And so my current research is pioneering the development of materials in which the fungal organism stays alive. The focus here is to study the regeneration behavior of the fungi, which leads to the self-healing of the material upon damage. So imagine that your pure mycelium leather handbag or shoe would self-heal after a crack or after a hole. And so with this research, we think we are taking an even larger holistic approach by extending the lifespan of an already biological material. And so we investigate the requirements to sustain the fungal viability during its lifespan. We are unraveling the correlation between the biological and the mechanical properties of the living material. And all of those insights are informing the design of new reactive and self-sustaining fabrics. The research is extending even more our knowledge about mycelium materials, because it's also focusing on the biology of the organisms, and it is taking the first steps towards the fungal engineered living materials in wearable and fashion applications. It's a very fundamental research, and my experiments are answering questions like, what is the infrastructure we need to enable the life of or the dormant state with a minimal intervention to enable the self-healing? How does the mycelium revive after it's being dried and post-treated? So what you see here is me cutting out holes in a dried and post-treated piece of leather, and do those new types of materials also keep their mechanical properties after their healing process. So I'm mostly interested in doing a multi-scale analysis from HIFA to application, where my role is to enable the healing process. I see I'm already at the end of my presentation. These are the acknowledgements of the different institutes I have or am working for, and also for my supervisors, because without their support and mentorships, all of this research would not have been possible. So I hope it was somehow interesting, and perhaps you have questions. I think we still have plenty of time to answer them.
Thank you. Thank you so much for this great presentation, Elise. You can hear us, right?
Yes.
Okay, great. So,
should I stop sharing my screen, or
however you wish?
Then you can see me better.
Okay. Are we on? Okay. Wait. Let me do this. Okay. So, just for in-person attendees, there is around 50 people on Zoom, 59 people on Zoom, and then we also have, I think, around 30 people here in person. So maybe we should start with the questions from the audience here. I already see some questions on chat, but if the Zoom audience, if you can keep writing your questions, we will address those after the questions from the audience. Okay. Any questions? Okay.
Hi, thank you so much for the talk. So one of the things that you had mentioned was, I mean, there was a lot of things you mentioned. They were all amazing, and I'm very interested in all of those things. But one that just wanted to get more detail on was you had mentioned that you had a substrate mixture of plastic and some other organic materials, and part of the interest was like intentionally degrading that material as like but it also, it has a second purpose as like a structural something. What was the plastic? Did you have to like treat that before you included that in the substrate, or was that something that you had to investigate when you were looking at this degradation mechanism through the fungi?
Yeah, we tested different types of plastics. So both soft and hard polyethylene. And basically what and we tested different types of species as well. All of them were white rot fungi, which are used in mycelium materials. And we, it's actually a research which I will present in a month. So there will be a research paper coming out quite soon about it, explaining all the details about the exact research method and so on. So have a look to that. But basically what we did was first making sure that the fungi was growing on the plastic. And without any other type of nutrients. And so what happens, what we think happened is that there was a kind of surface degradation. So there are, in plastic polymers, there are carbon linkages. And we think what the mycelium was doing, the fungal species was doing, is basically cutting those carbon links, allowing the surface degradation of the plastic. So fungal organisms on their own are not able to fully degrade plastic, but making it like basically being the first stage within the degradation of the plastic. Not sure if that answers your question. I can't hear anything now. Oh, yeah.
That was great information. Thank you very much.
Yeah, okay, great.
Sure. So I'm José Duarte, and I'm the director of SCDC. Elise, thank you so much for such an interesting presentation. I was taken by what you were showing. I have a question, from the architectural side. What would you say that are the biggest challenges ahead to, reach the level where this will become a standard application in architecture? What do you see as the biggest challenges?
There are many. I think most of it depends whether you're talking about a research challenge or more of a commercialization / scaling challenge, because those are, very separate things. Yeah, especially if you want to scale it towards a mainstream application, you need a large scale fabrication, meaning there would need more companies doing this. Probably, I'm not sure if the accessibility to waste stream fibers from a logistic point of view is so easy. So yeah, I think from what I see, but I don't have that much insight into what is actually happening into larger scale companies such as MicroWorks or Mogu or Ecovative. I'm more from the research perspective, but there is no bulk material at the moment. You can't go to the store. You can't actually order on a large scale those type of materials yet. That's mainly because it's not that old. It's about 10 to 15 years old, so it needs more time to grow, I think, into a larger scale fabrication. And then, yeah, I think we are seeing a shift from material composites, which are usually used for packaging or insulation, more towards leather materials, possibly because there are a lot of norms, there is a lot of regulation needed in construction materials, and we don't have those norms yet. So I'm not sure if companies are maybe struggling with getting those materials approved and then being able to scale up that process. And then from a research point of view, I mean, we have seen more scientific research in this domain going on, but I think it's still very much in its, in Belgium we say in its children's shoes. it's yeah. I don't know if I need to elaborate on what kind of.
I think it was a great answer. Thank you so much.
Okay.
Hello, my name is Maria Jimenez Gasco, and my question is, to what extent does the fungal species play a role in some of the mechanical characteristics you've seen, or is it, as long as it's a white rot fungus, it's more about the bioprocess, you know, the fabrication process, and the species doesn't really matter. I think most of the things you've shown were what, Ganodermas. So for a lot of these type of technologies, it's just a handful of fungi that are being used. And I mean, there's so many out there. So I'm wondering, what are your thoughts on that aspect? Thank you.
There are a million species, and the reason why we are taking just a few, like 5 main species, or about Ganoderma, Trametes, Pleurotus, like those are the main species that we see constantly in coming back in literature. Why? Because they do have interesting properties to bind substrate together, and not all fungal species have those properties. But I'm sure I'm not a mycologist, but I'm sure there are many more species to be found. The reason why we picked those is because they are commercially available, easily accessible. They are already well known into literature, so then it's easier to build up some knowledge. But it doesn't mean that other fungal species like brown rot, I've seen literature with brown rots and creating mycelium materials, it's also possible. So yeah, I think that's a really interesting research to still be done from a in a bio or microbiology department.
Thanks, Elise, for that answer. This is Zubeida Unais, and thanks, Maria, for that awesome question. The good news is we're going to talk about that exact point this afternoon in one of our panel, because that definitely is a question that we would like to either focus on or understand more as we go forward towards these working groups and forming the teams here at Penn State. So stick around. Any more questions from the room before we move on to Zoom? One more question from our friend in the back.
Hi, again. My name is Max Wetherington. I work in the materials characterization lab here. Yeah. I also had all of those things interest me. All these questions interest me. They're pretty broad questions. I'm very specific, detailed here. So the bio-weld, I was fascinated by that as well. Did you happen to do any kind of structural analysis on that, you know, that synthesis, or that after this weld was completed? What's like structural integrity thoughts on that? Yeah.
Yeah, unfortunately, I was not able to do it. COVID happened and the lab was closed. And so I had to wrap up my PhD, finish the writing, and the funding was over. So I couldn't do it. It is still something we will be researching this year. So now that I have funding again to continue research, I can do those tests and we'll do this summer. So I can't tell you anything about it, unfortunately.
Okay, well, thanks. Well, I'll be waiting with bated breath until we see what happens there.
Yeah.
Hi, my name's Jesse Bischoff. I was curious, I guess, on the flip side of the different species of fungus that you were using, the different sort of feedstock that you were using. Did you notice anything as far as, you know, a lot of people pick different woods for just the structural properties or the growth patterns or things like that? Were there many differences amongst the different wood types that you may have used, or was it mostly based on the nutritional levels to grow the fungal material?
So in terms of growth capacity of the fungal organism, or in terms of the mechanical properties of the final product?
Yeah, I'm more curious as far as the mechanical properties. Did you notice that maybe different wood material or whatever the base material was provided different outcomes despite having potentially similar amounts of growth within them?
No, the tests were not significantly different. So whether you use hardwood, beechwood fibers, or for example, hemp fibers, the mechanical properties will not be significantly different from each other. Well, it really then it depends on the property of the fiber itself. But I found that, well, in my limited experience of research, obviously there are many more researchers in this room doing similar work. But I found that the size of the fiber had a bigger influence on the mechanical properties, obviously because you're able to densify the material more properly. But in terms of chemical composition of the different fibers, there was not a big difference in mechanical properties if it was able to grow. Because, for example, the Trametes versicolor had a hard time to grow on softwood. And if it wasn't able to fully colonize the substrate, obviously the block would just fall apart more quickly. Yeah.
Thank you.
So it's really a combination of picking the right species with the right fiber and avoiding stress factors such as not enough nutrients.
Thanks, Elise. I think it makes sense to go to Zoom. I saw a few questions come in, and then we can come back to our in-person audience.
Okay, so we have, I think 7 questions on Zoom. I'll read these for the in-person audience too. The first question is, can this material be used, I think, to absorb CO2?
So fungi themselves do not absorb CO2, they produce CO2, they are more linked to humans. So when we breathe, we also breathe out CO2. The same is happening with fungi. So when we see CO2 sequestration used within fungal material domain, it's more because the fibers on which it's grown is a plant-based fiber, and that plant has absorbed CO2.
Okay. Thank you. Okay, and then the second question. How does the denser mycelium reduce the mechanical properties? Would a denser mycelium not improve, say, the compressive strength or tensile strength?
Um,
Were you able to hear, Elise?
yeah, I'm thinking about what the question, what is actually meant with the question.
I think you answered partially this question during your presentation when you showed the microscopic analysis.
Yeah. So the longer the material grows, the denser the skin will be. But at the same time, the longer the mycelium grows, the more it will degrade the fibers. So it's really finding an equilibrium in stopping the growth at the right moment. So yes, the density does improve the mechanical properties because it's holding the block basically more tightly together. But then at the same time, the properties of the fibers themselves reduce. And I think that has a more significant impact on the compression strength. I haven't done tensile tests on those specific experiments, so perhaps the tension would be improved, but there's something to be tested there. I don't know.
Okay, thank you. The next question is regarding the living leather. So would repair of dead leather be possible via re-inoculation with live fungi? So keeping your shoes alive and fed seems like a core. This is from Charlie Anderson from Penn State.
The exact methods that I'm using to keep the mycelium alive, I'm not able to disclose yet, so I won't talk a lot about it. But basically, yes, we kind of keep the shoe in a dormant state. And just like you're treating leather jackets, you would be able to treat, or your shoes, you're also treating them once in a while. So the idea is that if there is a micro crack or a larger crack, so we are able to heal up to 2 centimeters holes into the leather. Yeah, it would be like healing a wound. You need to perhaps apply a plaster or have a human interaction with it. So it's not completely self-healed, not completely autonomous at this point. Maybe it will be in 5 years' time, but who knows?
This is super exciting. Thank you. Another question, which I think you also answered during the Q&A from the audience, but they're asking which fungal species work best as biomaterials. Would you like to repeat? At least the ones that you used?
Yeah, so the species that I prefer to work with is Ganoderma lucidum. Again, it also depends a lot in with what kind of strain you're working, because within the, let's say, the category of species, there are hundreds different strains of Ganoderma lucidum, all with a different number, coming from a different country or a different location on Earth, and they have different properties. So even within one fungal species, I'm seeing very clear differences in growth patterns, in growth speed, and in mechanical properties. Yeah, because they behave differently. So it's really hard to say, oh, you need to use this species. Mostly Ganoderma lucidum, Trametes versicolor, and Pleurotus ostreatus are the species which are mostly used in literature. They are not necessarily the best.
We'll have a question from the audience.
Yes, thank you. My name is Merdad Hadigi. I'm head of architecture here. I have a question about your catenary, two questions actually about the catenary vault that you made. The first one is whether the knit material was purely formwork or also integrated structurally into the final outcome. And the second question is that structurally, the catenary form we can imagine working well. And the upside down catenary form as a vault, we can also imagine working well within simple confines of structures and forms. What I'm curious about is in the process of turning from the catenary form to the vault form, what did you have to do to maintain the integrity of the structure? Thank you.
Yeah, so two questions there, right? Whether the knit was structural or only a formwork. So it is the formwork during the growth, and it becomes the structural elements because the mycelium is stiffening up the knit. But we didn't, like it was made with linen and wool, and was knitted very strongly because knitting it's an elastic material. So we had a lot of problems in the beginning because it was actually, we couldn't keep the form because the tubes were deforming because of the elasticity of this knit. So we reduced the basically the knit size. We made it a double thread instead of a single thread. So it was a very hard knit, actually, very hard textile. And then indeed we had a lot of thoughts, like we spent different meetings in brainstorming how are we going to turn this structure without breaking it. Because indeed we knew that when we would turn it around, the upper part would then could break off. So we bought a huge balloon, which we wanted to put inside. But it's a kind of weather balloon, which has a diameter of 2 or 3 meters, which would fit perfectly. And then with the balloon, we would have turned the structure. But the balloon snapped. We had only ordered one. We had to turn the structure. So in the end, we just turned it by hand, basically, and nothing broke, nothing. There were no problems at all of the bending. And that is perhaps because there was still some kind of flexibility within the structure. We are not talking about masonry vault here. It's still textile, and it is the mycelium. So the textile has it can take up the forces in tension, and then the mycelium could take up the forces in compression, and that's why I think we didn't have much problems. Also because the dome is composed of tubes, so it's not fully like a fully grown structure, so that's why it could still bounce a little bit while we were turning it.
Elise, thank you. We have a couple of more questions from the chat, but I also have a follow-up question about the Bionite project. So we also have a very similar research project here together with Felicia Davis and her PhD student Farzana and my PhD student Ali, called Myconit. So I think we while we were working, you guys also made the beautiful Bionite project, and we were okay, so this is doable. Let's we also, that also encouraged us. So I have a question about you mentioned that since it's very large scale and it's not component based, that you were also afraid of contamination, which is a challenge when you're working with mycelium based materials in large scale. And you said you systematically like cleaned the contaminated parts. Can you maybe explain that a little bit? And one more question about, so I believe then you kept the structure alive, right? Because it's too large to be dried in ovens. So is it still alive, or are there fruiting bodies on the structure? Can you talk a little bit about that too?
Yeah. So the contaminations, we have taken slides. I don't know what's the right word in English, but we've scraped off the contamination and then put it into the freezer to be able to analyze which species exactly were growing on there. But we saw a green type of contamination and the black ones and some pink ones. So there was all kinds of environmental fungi growing on there, which is normal. It's a building where the door is opening constantly. We were not able to close off anything there. So we did expect having it. And we debated a lot whether we needed to take them off from a health purpose or not. I don't know in the United States if health and safety is such a big thing as it is in the UK, but we have to fill in forms for health and safety, and it has to be properly removed. Otherwise, we were afraid to have to throw away the Bionite completely. So that's why we basically were like, from the moment they occurred, we put an antifungal spray on there, and then ethanol, 100% of ethanol, and then taking that part off, which meant that on those specific locations, the fungal, the mice, like the Ganoderma lucidum, which we used here, was also not able to grow. Although there were some parts where the Ganoderma species was able to overgrow the contamination, but it didn't happen everywhere. It would have been interesting to have a multi-species kind of structure, allowing all of them, but yeah, that's why we got rid of it. And then the second question, I forgot what it was.
It was about, is it still alive?
Yeah. So the structure is air dried, and it's fully air dried. So we had some parts, some testing element growing in there, which we could take away and then cut open, and we noticed everything inside those testing pieces was fully dried out. And by air drying, you mostly also, you could be able to revive potentially the organism, but you need a lot of humidity to kick it off. We had tiny fruiting bodies, but like really one or two, not very significant.
Thank you. A couple of questions from chat again. Okay. Wait. So some of these questions are already addressed, but with your answers for the questions from the audience, I would say. Okay. I mean, this connects back. This I think connects well to the question that you just answered. If the mycelium becomes alive again to repair the material, would fruiting bodies form, releasing potentially harmful mycelium spores, or is this only if it's feeding on substrate instead of being pure mycelium?
So to get fruiting bodies, you also need specific conditions, and those conditions are not there with pure dried leather. So the fungal organism mostly create fruiting bodies as a stress factor to reproduce at the end of its growth phase. But I haven't seen this on leather yet because the fabrication method is completely different. So to create the fruiting bodies, it will still need nutrients like wood fibers, for example, to create them. So I don't think that's a big challenge, but it is one of the questions that we are trying to answer. Either you could approach it from an engineering point of view and engineer the species so that it doesn't produce the fruiting bodies, or else from a chemical point of view and add chemicals so that it also doesn't produce the fruiting bodies. But it's not the biggest challenge. Like it is one of the questions that definitely will come up when going towards the application. But at this point, it's not the main. It's not part of the main problem. Yeah.
Thank you. And one last question, and that's about using and building with these materials in outdoor conditions. Is it possible? And how does the climate or seasonal changes affect this kind of fabrication?
Yeah, it's a good question. Actually, the only research that I've seen about it is coming from your group. So it's definitely a challenge. I haven't addressed it myself yet, so I think you guys are better to answer this question than I am. Yeah.
I mean, I can answer. We just recently participated in a biomaterial building expo at University of Virginia, and we built an outdoor structure. And we had a student who monitored the process for a month and a half, right? A month and a half, they took pictures, and now that they disassembled the structure, they're gonna ship it back to us so we can analyze what are the changes in the components. And we're gonna compare it with the component that we kept inside. But I could tell that based on what the student from the University of Virginia told us, it was holding its shape. There were some bird poop on it, so birds liked it for some reason. But other than that, they said it held its shape fine. But we need to look at the material when they ship it back to us to see. But I think your answer, yes, there's not much research about how these materials or what kind of changes happen to these materials when they're subject to outdoor conditions and environmental changes. So that's a possible line of research.
Mm-hmm. Yeah, and even indoor conditions, like I've seen many of my samples that I just kept into my closets being eaten up by insects. So if they are not dried, they are lovely food for any kind of other organism to feed on it. Yeah.
Yep. Elise, thank you so much.
You're welcome.
So,
thank you so much as well.
For Zoom audience and for also in-person audience, we'll give a short break and we'll continue with the panel. Elise will also be a panelist. She will be joined by Juan Pablo Gebaudan and Esther Obonio in this panel on sustainable built environment. So we're going to give a short break and we'll reconvene in 7 minutes.